All right, everyone, welcome back. We are now at the fourth lecture in the blood chapter, and today we're going to be, again, covering just a little bit to do with erythrocytes, the red blood cells, and then some of the proteins that are in the blood as we talk about blood typing. Now, this will be an important lecture because it will also overlap with one of our labs where we cover blood typing. So kind of use this as your lecture and lab material this week. As always, let's go ahead and start with our not really attendance questions, or in this case, question. What are alpha, beta, and gamma globulins? Go ahead and try to answer those on your own. Okay, hopefully you've done that. So let's see. What are alpha and beta globulins first? Let's cover those together. Those, and we covered all of these during the plasma area, where we talked about proteins in the plasma. So if you don't remember these, go back and refresh on that plasma lecture. Alpha and beta globulins are transport proteins. And gamma globulins, that's another name for antibodies. And today we will be talking a little bit about antibodies, and then we will talk more about them when we get to the leukocytes and immune system. So today we're going to be talking about blood groups, and I want to talk a little bit about some terms, and then we're going to do some drawing. So when we talk about blood groups, we hear terms like, I'm blood type A, or blood type B, or blood type O, or blood type AB. And then sometimes you'll also hear positive or negative. So maybe someone will say, I'm type A positive, or I'm type B negative. And when we talk about blood types, really what we're talking about is, what do we have on the surface of our erythrocytes? That's what blood types are telling us. It's telling us about what types of proteins we have on the surface of our red blood cells. Now, when we talk about those things on the surface of our red blood cells, think back to chapter 3 when we talked about cells in general, and specifically talking about the plasma membrane. Remember when we were looking at the plasma membrane and all of the stuff that we find on the surface? Well, there were a lot of proteins and glycoproteins, things like that. So when we talk about the blood type, we're really talking about those proteins that are on the cell surface, and specifically we are going to use a term, antigen. So when we see the word antigen today, we are talking about those proteins that are on the surface, but specifically on the surface of our erythrocytes. Now we will see the word antigen quite a bit for the rest of this chapter. And when we see it, it's just talking about those surface proteins. Now, when we get to the immune system, we will see antigen used a little bit differently, and it's kind of being used to mean foreign antigen. So let's talk a little bit about antigen and foreign antigen before we get too deep in the weeds here. Antigens are on the surface of all cells, your own cells and other cells, other people's cells, other animals' cells, plant cells, bacterial cells, all of those things, they will have antigens. But our immune system uses those antigens to determine if something is supposed to be in your body or not. And it does that by looking at the shape of those antigens. So really what it's doing is determining this cell is you, or this cell is not you, because of the shape of those antigens. The antigens on your cells are shaped similar, but not identical to any other living thing. No two living things, unless they are identical twins or clones, will have the same shaped antigens. So our immune system is constantly going through and looking at the shape of the antigens on the cells in your body. And if they are shaped correctly, that's great. They belong there. If they are shaped incorrectly, that triggers an immune response. So that's going to be a little bit of the foundation for both what we talk about today and the immune system. So antigens, those are the proteins on the surface of your cells. And like I said, later we will see self-antigen. Those are the proteins on your cells. Or foreign antigen. Those are the antigens on cells that should not be in your body. Now, the other thing that we're going to see today are antibodies. Remember, antibodies, also called gamma globulins, they float around in your plasma. We will see where they come from in another lecture or two. But antibodies, they are proteins in your plasma. And the way antibodies work, these are some of the parts of your immune system that look for those foreign antigens. And they do that based on their shape. We will see how when we start drawing. So antigens are on the surface of your cells. We are going to be talking today about some special antigens that are found on your red blood cells. And antibodies are found floating around in your plasma and are part of your immune system. Now, there are some other terms that you may see now and then. I will use antigen and antibody, but your book also introduces some other words, and you may see some other words in homework. And that is agglutinogen, which is another name for antigen, and agglutinin. Agglutinin, that is another name for antibody. Agglutinogen, antigen. Agglutinin, antibody. Like I said, we use antigen and antibody. Much more straightforward. So let's do a little bit of drawing and see how blood groups work. So first, I'm going to draw a red blood cell. And on the surface of that red blood cell, there are going to be some proteins. So when I draw today, I'm going to use some different shapes. I'm going to use square on a stick, triangle on a stick, and circle on a stick. When I draw those, they will be exaggeratedly big, just so we can see them. But when I draw them, what I'm actually drawing are antigens. And I'm going to use the square shape to represent antigen A. I'm going to use the triangle shape to represent antigen B. And I'm going to use the circle shape to represent antigen RH. For right now, we are only going to be talking about A and B. So, here is a red blood cell, and let's say on the surface there are lots and lots and lots of proteins shaped like square on a stick. And we see over on the left there that is antigen A. So this red blood cell has antigen A. I only drew one, but it would be covered with them. The way that we indicate this, the way that we say this person's blood cells have antigen A on their surface, is we say this person is blood type A. Blood type A says this person has antigen A on the surface of their red blood cells. All right, so let's draw something else. And again, right now we're ignoring Rh, so we didn't say positive or negative. We'll come to that later. Let's say we look and we see this person has triangle on a stick as their antigen. So they have antigen B. The way that we say this person has antigen B on their red blood cells is we say this person is blood type B. So what we're seeing now is blood type is the antigen that this person has. When someone tells you their blood type, really what they're saying is the type of antigens I have on my red blood cells is. So let's try another one. Let's say we look and this person has a lot of antigen A on the surface, but they also have a lot of antigen B on the surface. They have both. So we could say this person has antigen A and antigen B. So this person is blood type AB. There is no antigen AB. What we're saying is this person is blood type A and blood type B at the same time. This person has both A antigens and B antigens, so they are blood type AB. And one final one to look at. Let's say we look and this person has no antigens at all. And really, that's not true. We have other types of antigens. We are just specifically talking about blood typing antigens. And we see that there is no antigen A on their red blood cell surface. There is no antigen B on their red blood cell surface. So what we say is this person has 0 antigens. Another way to say that is this person is blood type 0. But historically, it didn't come across that way. When we were labeling the blood types as they were being discovered, we called some of them blood type A because they had A antigens. We called some of them blood type B because they had B antigens. We called some of them blood type AB because they had A antigens and B antigens. But you never hear of someone being called blood type 0 because they have 0 antigens. Well, what actually happened was there was some weirdness and someone saw all these letters, A's and B's and AB's, and they thought that that 0 was the letter O. And that's what stuck. Blood type O actually means blood type 0. Blood type O tells us there are 0 antigens on their erythrocytes. So blood type O, no antigens on their erythrocytes. There is no antigen O. Antigen O just means no or 0 antigens. Okay, so now let's grow with that a little bit. Let's bring in the other side of things. Well, right now, let's condense this blood type. And let's go back to the person with blood type A. We said if they have A antigens, they are blood type A. There is never a time when someone with blood type A should have B antigens in their bloodstream. If cells with B antigens shows up in their bloodstream, that means that something has gotten into their body that shouldn't have. And it is the job of our immune system to monitor for things that should not be in our body. One way that that happens is with antibodies. Gamma globulins. The way antibodies work, well, they're these branched proteins. Some of them will have 3 branches, some will have 5, some will have 7. I just draw 3 branch for ease. Now on the surface, or at the end of each of these branches, will be a receptor. And these receptors will have all kinds of shapes to them. We have lots of different antibodies with lots of different shapes. But the shapes are always going to be shapes that should not be in our body. That's how they work. They look for things that should not be in our body. They do not look for things that should be in our body. So, there should never be antigen B in our body. So, our antibodies, some of them will be shaped so that they would fit against antigen B. Here we can see this little triangle receptor, and another triangle receptor, and another triangle receptor, shaped to fit the triangle shape of antigen B. If there were to be an antigen B that shows up in our body, it would have these antibodies stick to it like a puzzle piece. They would snap together, and then one of those antigens from one cell would stick into this receptor area. Another red blood cell with an antigen B would stick here, and another one would stick here, and all of these blood cells would start sticking together in a process called agglutination. Agglutination is clumping. Now, that is not the same as clotting. Clotting is something different that we will learn about on another day. Clumping is called agglutination. And when this happens, one, it's causing all of those cells to stick together. And two, it's going to cause another reaction that results in these cells being split and broken apart in a process called hemolysis. Hemo for blood, lysis for splitting. It's going to break these cells down, hemolysis. That happens when an antibody finds an antigen that should not be in your body, in this case, an antigen on a blood cell. So someone with blood type A would have anti-B antibodies, because antigen B should never be in someone with blood type A's body. Let's do another example. So let's say we look and someone has triangle antigen. What is their blood type? Their blood type is blood type B. What antibodies would be in their plasma? The ones shaped to fit antigen A. They would have anti-A antibodies. Whatever their blood type is the antigen that they have on their red blood cells, or another way to say that, the antigen that is on their red blood cells is their blood type. So blood type and antigen are always the same. Now, antibodies are always the opposite whatever their blood type, they should have the other antibody. Blood type A has A antigens and anti-B antibodies. Blood type B has B antigens and anti-A antibodies, like we have drawn here. Let's do just another example or two to make sure that this sticks in before we add to it. What if the person has both A antigens and B antigens? What is their blood type? This person is blood type AB. Again, that does not mean they have something called AB antigen. There is no such thing. This means they have A antigens and B antigens. So what antibodies would they have? Would they have anti-A antibodies? No, because they've got A antigen. Would they have anti-B antibodies? No, because they've got B antigens. They would not have any antibodies. And again, that's not true. There are other antibodies. We are just talking about the ones to do with blood type. So we say antigens A and B is blood type AB. No antibodies in blood type AB because all of those antigens should be there. There's nothing to look for that would be out of place. And finally, what if we look and this person has no antigens? Well, what did we say? They are blood type O. What antibodies would they have? Should there ever be any A antigens in their blood? No, there shouldn't. So they would have anti-A antibodies. Should they ever have any B antigens in their blood? No. So they would have anti-B antibodies. They would have anti-A and anti-B antibodies because A antigens and B antigens should never be there. Okay. So that is the ABO blood system. But there are other blood systems. The one that we're going to talk about next is the RH factor. Now, RH is just another antigen. So let's draw this out using both the ABO blood group and the RH blood group. Let's say we look and this person has A antigens and RH antigens. What blood type are they? Well, they have blood type A because they have A antigens. They do not have B antigens, so there is no blood type B for this person. They do have RH, so it's tempting to say this person is blood type ARH, but that's not what we say. When we talk about the ABO blood system, we just put down the antigens that they have. But when we talk about the RH, we say it differently. We say that this person is positive for the RH antigen or the RH factor. The positive means, yes, this person does have the RH antigen. This person is blood type A positive. Let's do another and say instead, let's say this person has triangle on a stick, the B antigen. So what blood type is this person? This person is B positive. They don't have A. They do have B. They do have RH. So they have B, blood type B, and they are positive for RH. Positive, all it means is yes, this person has the RH antigen. Now, using someone with blood type B, what antibodies would they have? Should they ever have a antigen in their body? No. So they will have anti-A antibodies. Should they ever have B antigen in their body? Yes. So they will not have anti-B antibodies. Should they have RH antigen in their body? Yes. So they will not have RH antibodies. All right. So now, again, still what we're seeing is whatever antigens they have, that's their blood type. Whatever antigens they don't have, they will have those antibodies. Now, final one, and this is where it gets tricky. Let's say we look, and this person has A antigens. So now we're still doing the ABO and RH. So if this person has A antigens and they do not have B antigens and they do not have RH antigens, what do we say? Well, they are blood type A because they have A antigen. They are not blood type B. They don't have B antigen. And they do not have RH. So we say they are negative for RH. Blood type A negative means they have A antigen and they do not have RH antigen. The negative just means they do not have RH. There is no positive or negative RH antigen. There is either RH antigen or not. RH antigen, you put a positive sign. No RH antigen, you put a negative sign. Positive means they have it, negative means they don't. Now, let's talk about antibodies in someone with A negative blood. If they have A negative blood, should they ever have A antigens? Yes. So they do not have anti-A antibodies. Should they ever have B antigens? No. So they will have anti-B antibodies. Should they ever have RH factor? No. So, weird thing about the antibodies in this situation. When someone is born with blood type A, they are born with antigen A on their red blood cells. And they are born with anti-B antibodies. When someone is blood type B, they are born with B antigens on their red blood cells. And they are born with anti-A antibodies. When someone is RH positive, they are born with RH antigens on their red blood cells. When someone is Rh negative, they are not born with anti-Rh antibodies. There's a different mechanism here. So blood type A negative has A antigens on their red blood cells and anti-B antibodies in their plasma. But they are not born with anti-Rh antibodies, and they could potentially live their entire life without making any anti-Rh antibodies. But if they ever come into contact with Rh positive blood, for whatever reason, maybe they have a blood transfusion that's incorrect, maybe they do the thing that little boys used to do a long time ago and do blood brothers. They cut their thumbs and rub them together for who knows what reason. Whatever happens, if this person comes into contact with Rh positive blood, then from that point forward, their body makes anti-Rh antibodies. And they will have those anti-Rh antibodies for the rest of their life. But they were not born with them. We say they had to be stimulated to make them. Now, why is all of this even important? It's because of blood transfusions. So let's go back to our PowerPoint and talk a little bit more, and then we will see when it's okay to give a transfusion. So we said that blood groups, the Rh factor is different from ABO. Now, it plays a big role in pregnancy. We'll see why. Because we run the risk of something called erythroblastosis fetalis, or hemolytic disease of the newborn. But what's the mechanism that causes that problem? When we give transfusions, we have to make sure that it's safe. Who can give to who, or who can receive from who? Because you don't have to have an exact match. A negative to A negative, or B positive to B positive. Yes, those are safe. But there are some others that are safe also. So what is safe and what isn't? What it comes down to is what would cause agglutination. And to make sure you don't cause agglutination, you want to make sure that you never introduce a foreign antigen. You can put blood into a person safely even if they don't match exactly, as long as you do not introduce a foreign antigen. Because if you introduce a foreign antigen, you get agglutination. And that's what we see over here. Agglutination is when the blood starts to clump together. So let's go back and do a little bit more drawing. And say someone with blood type A negative. Who can they donate to? Well, what antigens do they have? Do they have A? Yes. Do they have B? No. Do they have Rh? No. So, if this person is the donor, well, the recipient needs to have antigen A. This person, the recipient, can be blood type A negative because that's an exact match. The recipient can be AB negative because they already have A, and they are negative for RH. The person receiving can be a positive because, again, A antigen is okay. And if they don't have RH, if they are negative, we're not introducing anything. And AB positive. A negative donor can donate to a person with A negative, AB negative, A positive, or AB positive, because in all of those instances, we are not introducing a foreign antigen. What about somebody with A positive? What antigens do they have? Do they have A? Yes. Do they have B? No. Do they have RH? Yes. So this person can donate to anybody that has both A antigens and RH antigens. Can they donate to A negative? No, because the A negative does not have RH antigen. They can donate to A positive. They can donate to A B positive. And that's it. Because anything else that they donate to, they would be introducing a foreign antigen. They would either be introducing A or they would be introducing RH. But let's talk about a special situation. Let's talk about pregnancy. If mom is RH negative, she should never have a situation where RH positive is in her body. If mom is RH negative, she should never have RH antigen in her body. Well, let's say dad is RH positive. Well, it is possible for them to have a baby that is RH negative. Let's call this baby number 1 is RH negative. Mom carries the baby, goes full term, gives birth, and it's just fine. Because baby does not have RH antigen. Let's say baby number 2, well, he gets a little bit more from dad. He is RH positive. Well, mom carries the baby, full term, no problems. But during delivery, well, delivery can be a little bit traumatic, and there is some bleeding involved. And during that delivery process, mom is going to become exposed to baby. Mom's blood and baby's blood are going to intermingle. From that point forward, since an RH negative mom was exposed to RH positive blood, what's mom going to do? Mom is going to start having anti-RH antibodies. Well, baby number 2 was born and he's just fine. But baby number 3, let's say he is also Rh positive. Now we have a problem, because at this point, mom has anti-Rh antibodies that will recognize baby 3 as foreign, and her antibodies will bind to baby 3's blood and cause agglutination and hemolysis. Remember, we call that erythroblastosis fetalis, or hemolytic disease of the newborn. Erythroblastosis fetalis, or hemolytic disease of the newborn. That is when mom is Rh negative and has previously delivered Rh positive baby. So it would be an issue for an Rh negative mother on her second or later Rh positive baby. It would not be a problem with the first Rh positive baby. It would not be a problem for an Rh negative baby. And it is never a problem if mom is Rh positive. It is only a problem when mom is Rh negative and mom has previously delivered an Rh positive baby. It is a problem for the next Rh positive baby and everyone after that. So, what do we do in that case? In that case, mom is given something called RhoGAM. RhoGAM is something that blocks mom's anti-Rh antibodies to prevent that reaction from happening. Now, even though we say it is only for an Rh negative mother who has previously given birth to an Rh positive baby, there's a little bit more to it than that. If you are an Rh negative mother, you are going to be given RhoGAM, regardless if you have given birth or not. So this is why it's so important for pregnant mothers to get those early exams. As soon as you find out you're pregnant, you schedule a visit. And if you are Rh negative, the doctor is, well, one, they're not going to say, have you given birth to an Rh positive baby before? They're not even going to ask if you have ever come into contact with Rh positive blood in any form. They are just going to assume that you have, for so many different reasons. One, maybe you didn't even know you were pregnant at some point and miscarried without even realizing it. Or maybe you are wanting to cover something up, or whatever the reason. The doctor's not going to wait to get a history. If the doctor realizes that mom is Rh negative, they are going to give RhoGAM to mom. And that is going to prevent erythroblastosis fetalis, hemolytic disease of the newborn. It's a safety measure. So, one last thing, and that is something that is on the way, and it's actually started to already be rolled out a little bit. And it's something created by the University of British Columbia. It is an enzyme that, if you put it into blood, it's going to cleave all of the antigens off of all of the erythrocytes. Why is that important? Let's use that as a thinking cap question. What if an enzyme took all of the antigens off of all of the erythrocytes? Why is that a good thing? Well, remember, what do the antigens do? They determine the blood type. So if we take all of the antigens off of the erythrocyte, what blood type do we have? We have O negative. O because there are no A's or B's. Negative because there are no RHs. So O negative. And what's important about O negative? It's called the universal donor. It can be given to anyone. The reason behind that is because it does not have any antigens. It is impossible to introduce foreign antigens if there are no antigens there at all. That is why O negative is the universal donor. On the other hand, AB positive, by the way, that's the universal recipient. They can receive from anyone because they have all of the antigens. It's impossible to introduce a foreign antigen to someone who already has them all. But here, O negative blood is created by this enzyme. And the benefit for that is now you can give it to anybody. All right, so that brings us to the end of this lecture. Next time, we will start talking about white blood cells and the immune system. In the meantime, if you have any questions at all, reach out, and I will talk to you in the next lecture.